Okay. Hello, I'm Vanessa Jones.
And I'm Lacey Mims.
And today we're going to be talking about our own personal experiences doing undergrad research and some of the roadblocks we've encountered and how we've overcome them in regards to our projects. It's easy from an outside perspective to look at research projects and think that they're a pretty straightforward linear process. Like we read primary literature and we learn about these pivotal experiments in biology or chemistry, and they all seem to kind of follow the same formula, where you come up with your experimental question, you perform your experiments, you get viable results that you interpret, and then you present your findings. But what you don't see are all the years of troubleshooting these experiments and working past these roadblocks that are very typical to the process. So it's not the case that it's this easy, simple, linear process. And that's kind of what Lacey and I are going to address today in our presentation, that in our personal experiences, the results of our experiments have gone off track from what we expected. But we're also here to talk about how we've dealt with that and what we've taken away from it.
So given that this isn't really a typical research presentation, we wanted to give just an overview of the flow for today. So we'll start out by giving a very brief and broad background to our research, including research questions and our genes of interest. Since this is so brief and broad, please don't sweat it if you're not quite getting all the science behind the research that we're doing. We'll then move on to our general methodology and gene-specific research, including our kind of different troubleshooting that we did with the knockouts, and then we'll move on to our final takeaways from just doing undergraduate research itself.
So the background for both of our research projects is RumA’2B, and it is a DNA polymerase that is encoded by an integrative conjugative element, ICE391. And this polymerase performs translesion synthesis, meaning that it can repair DNA past lesions in the strand, which is significant damage. But as it replicates this DNA, it is prone to making mistakes. And these mistakes lead to mutations in the host cell, which can lead to things like antibiotic resistance. And that's why our research is particularly relevant. One interesting thing that we've noticed working with this polymerase in the lab is that when you express this polymerase within the genetic context of ICE391, we observe low levels of mutagenesis. But take this polymerase out, express it on its own in a plasmid, and we see high levels of mutagenesis. ICE391 encodes regulatory genes that affect the expression and affect the activity of this polymerase.
So when we're talking about kind of effects of protein active and regulation of protein activity, we're really looking at it from three different levels. At the DNA level, we have to ask, like, how often are these genes getting expressed? Then at the RNA level, how stable is this RNA transcript? How long is it going to stick around in the cell and be able to be translated? And then finally, at the protein level, we have to ask, is this polypeptide chain need to be modified in any way before it becomes an active protein? And how stable is the active protein within the cell's environment? When we're looking at the regulation of RumA'2B mutagenic activity, there's two additional lenses that we have to apply. So there's universal regulators that are present just within the E. coli itself. And then there, like Vanessa mentioned, there's also regulatory genes encoded for by ICE391 itself. And some of these universal regulators that come from the E. coli background itself include genes that are involved in the SOS response, as well as ATP-dependent proteases. Then within ICE391, we have a couple of negative transcriptional repressors, including SETR and CRO-S, as well as a hypothesized protease known as LON391. But even when we take into account the combined mechanisms and activities of all these different regulators, we still can't account for the total repression of mutagenic activity when RumA'2B is expressed from within the context of ICE391. So that leads us to ask the question, what other genes could be playing a regulatory role?
My gene of interest is ftsH. And it is one of those, we're looking into its potential as a universal regulator, meaning that it's encoded by E. coli itself and not ICE391. It is one of 5 ATP-dependent proteases in E. coli. The other 4 have been previously knocked out in this lab. This is the last one. And it's the last one because it is an essential protease, meaning if you knock ftsH out of a wild-type E. coli cell, that cell will die. So that is one obstacle that we've had to overcome with this particular gene knockout. From a functional standpoint, ftsH plays a few roles, but one of its main roles is that It helps indirectly regulate membrane composition in terms of the ratio of lipopolysaccharides to phospholipids.
So my gene of interest was Epsilon 391. Epsilon 391 comes from ICE 391 itself. And when we look at the sequence for Epsilon 391, we see that there's a lot of sequence homology or sequence similarity with Epsilon subunit for DNA polymerase 3. And the Epsilon subunit for DNA polymerase 3 gives this polymerase proofreading capabilities. So based on the sequence homology, it's hypothesized that Epsilon 391 may provide limited proofreading function for that RumA'2B mutagenic DNA polymerase. So now that we have these genes of interest and we have these hypothesized functions, how do we determine if these genes are actually doing what we think they're doing? And we do that in this lab through gene knockouts. And again, for this next section, if you're not quite getting all the science, please don't fret. This is really brief and broad. So in our lab, we do gene knockouts through a one-step PCR knockout process where we amplify out an antibiotic-resistant marker flanked by sequences of homology that correspond to our gene of interest. We'll then electroporate our cells to get that linear DNA into the cells. A recombination event will switch out our gene of interest with the antibiotic resistance marker. And then finally, we will loop out the antibiotic resistance marker, leaving us with a very small scar region where our gene of interest once was. Really simple, really straightforward, 4 easy steps, right? Not quite. So those 4 steps actually break down into about 17 other steps. And that's 17 steps where things can go wrong and you have to troubleshoot. And as you will soon see, it gets really complicated really quickly. And even when you have the knockout done, in order to test for the effect, there's additional analyses that need to be done. So now we'll move on to our actual gene knockouts.
So for my gene, ftsH, it was the actual knockout in and of itself that was unsuccessful. It was that electroporation of the antibiotic resistance cassette that was going to flip out the gene that did not work. Electroporation involves running a high voltage over the membrane of a cell to facilitate the uptake of linear DNA. And this process was successful most of the time and results in transformants. But at the same time, as you can probably imagine, when you're running a high voltage across a cell membrane, there's a possibility that the cell will die and not take up DNA. So while electroporation is good and effective and it produces transformants the majority of the time, it also has a generally low viability, cell viability. But when I performed this electroporation, I got no transformants. My time constant for the electroporation was very high. It was always coming out to 6.1 milliseconds when a successful electroporation usually comes out to 5 milliseconds. So something went wrong and we needed to troubleshoot. So we went through each element of the electroporation. Was the electroporator working properly? Was it out at all of the right settings? Was our plasmid fine, uncontaminated at the right concentration? Were our plates the proper antibiotic? Were our plates fresh enough? Were they contaminated as well? Was there a problem with my electrocompetent cells? Were they not washed well enough? They had too much solute in them. That was what was raising my time constant. So I remade those cells and redid the protocol as many times as the things that I have just listed off, which was many, many repetitions of this same protocol. And we were still getting the same results, no transformants. So Dr. G and I had to sit back and think about the background strain for this knockout. Because like I said, ftsH is an essential protease. So when you knock it out of wild type E. coli, that cell will die. So we had to prepare a specialized background for that strain so that it could survive without ftsH. And this background is an sfhC suppressor mutation. The problem with this mutation is that it alters the composition of the outer cell membrane. So when we electroporated and ran that high voltage across the cell membrane, we hypothesized that this altered membrane composition was just too weak to sustain that sort of electric shock, and that it collapsed in on itself, killing all of the cells. So in terms of the gene roadmap, gene knockout roadmap, this is as far as we got. We did all the preparatory work, but couldn't actually get the knockout. But it took from September of 2019 to the present to get to that stage, partially because of COVID, but also partially because of all the repetition that we had to do of that protocol to figure out what was going wrong.
So now looking at my knockout for Epsilon 391, there were quite a few roadblocks that I ran into, but as opposed to going to the specific roadblocks, I kind of wanted to go into more the conceptual framework for how I worked through when protocol didn't work out as it should have. And so every single time I don't get results from an experiment, I kind of ask myself, first and foremost, did I mess up my protocol? I review the protocol that we have, I look at my notes from the experiment and make sure that everything matches up. If everything looks good, I then look at the reagents I used. I make sure the reagents weren't contaminated, that they're within date and at the proper concentrations. If the reagents look good, then we look at the equipment itself. Was the equipment functioning correctly? And that's everything from pipette men to scales to our incubators, like Vanessa mentioned, our electroporation machine, and even the machine that we use to take the DNA concentrations that we use. If the equipment looks like it's going well, we then look at the protocol and we make sure that the protocol that we're using is consistent with the protocol that's present in the literature. If it's not consistent with the literature, we ask, do we need to change our protocol to better fit the literature? But most of the time it's pretty consistent with the literature. So then we all have to kind of sit down as a lab with Dr. Gonzalez and say, what else could be going on to make this protocol not successful? But one thing you should really get from this graph is that, there's a lot of repetitions and just tweaking the protocol a little bit to try to get it to work. A lot of times that we had to redo the protocol. And that's exactly what you see with the Epsilon 391 knockout. Had to prep out the linear DNA multiple times, had to remake my recombinant cells. I had to do multiple electroporations before I got a successful one, and even had to test out the machines and cells multiple times. Once we had a successful electroporation, it took me multiple times to kick out a certain plasmid we had in the strain, and then I had to perform multiple colony PCRs to just confirm that we had the knockout. But even though we had to go through this so many times with so many tweaks, eventually I was actually able to get the Epsilon 391 knockout and start testing for the effect of the knockout using spontaneous mutagenesis assay. So when we look at the results from the assay, we see that in the Epsilon 391 knockout, there was actually a decrease in mutagenic activity for that RumA’2B polymerase. And this effect is the opposite of what we hypothesized would happen. So, there's even additional work that needs to be done in the future to just figure out what Epsilon 391 is even doing. But, to get to this point, I've been working on this research project since November 2019. It took me 14 months to get the gene knockout itself. That's a lot of time. And, for the past few months, we've just been trying to analyze the effects of the knockout. So science takes time. It's a lot of tweaking and it's a lot of time, and that's just the bottom line. And that kind of leads us to some of our main takeaways. So one of my main takeaways from doing research as an undergrad is that no one has all the answers with research, and collaboration is key. From that, diagram, Vanessa and I have touched on it a lot. There's a lot of communication and trying to figure out why protocols aren't working, both with consulting the literature, consulting other lab students, but especially working with Dr. Gonzalez to kind of work through what we think is going on and coming up with solutions for moving forward.
And the second takeaway is you may not get the results you anticipated, but that's okay. It could be like Lacey's project where the unanticipated results take your project in a completely different direction than what you expected. Or it could be like the lack of results from my project where it tells you that you've hit a dead end and you need to stop. You need to stop your project. You've hit a dead end. And that is by no means a waste of time or a failure to not get results that are perfect, because science isn't perfect and it's more challenging to work through these problems. And that's really what real research looks like. And we'd like to thank Southwestern University, our scope donors, Antonio Mendez and Athena Pinero for their contributions, and of course, Dr. G.
And now we would really love to open up the floor for questions about the science that we did and our experience itself.
I had a question actually. Oh wait, you guys are raising hands. I'll wait my turn.
Okay. I think I saw Lexi's hand come up first.
I had a question. You said that you guys can look at your reagents and see if they're contaminated. What is the process of doing that? Like, how do you determine if it was contaminated or not? And do you just hold on to the reagents until you're done, or how does that work?
Yeah, so we'll hold on to the reagents, and also a lot of the things that we're using are nutrient-rich media. And when you're working with bacteria, you can kind of swirl around that media. If if you see stuff floating around, there's probably something growing in it, and you need to toss it out and redo it. And it's frustrating, but that's kind of how we look for contamination in that. Dr. Zwale?
Lacey and Vanessa, I loved your story. I'm sure my research students can totally relate. They're nodding. I would love a copy of your flowchart of how you troubleshooted to give to students. I think it really nicely outlined strategy on troubleshooting, because that's what research is. I do have some science questions, Vanessa. So I know that when you're doing these transformations with linear DNA, it's often more challenging than using plasmid, circular, like supercoiled DNA. So did you try that as a control just to make sure you could get supercoiled DNA into the cell? Because I can't even imagine linear DNA.
Right, yes, we tried the electroporation with a plasmid instead, and that did not work either. And a plasmid that we knew like was good and worked. So
Very good.
part of the troubleshooting process. Yeah, that's a good question.
Okay. All?
Yeah. So my question is towards Vanessa. In your experiment, by the way, both of you guys did an amazing job, and I really enjoyed your presentation. By far, probably my favorite presentation today. So my question was that, like, you were talking about how forgive me because I don't know the lingo as much as you guys do, but you were talking about how you added some component that made the membrane like weaken, and that therefore it wasn't able to being used. If you know what I'm referring to.
Yes, I do.
So is there any way of adding a secondary like component or something like that that would like alleviate that effort, or would that also complicate the actual like experiment?
Well, that's the thing. I'm not entirely sure what can be done to change that, because I know that that particular suppressor mutation suppresses a deacetylase that would otherwise be regulated by that protease. So I'm not sure exactly how to work around that issue, because all I know is that it's keeping the cell alive, and that was step one, the bare minimum. But beyond that, I've looked at the literature and I don't see much about adding extra things to that sfhC mutation. But I wish. That's a good question.
Thank you. It was really interesting.
Awesome. Ethan?
So Vanessa, kind of tailing off of that, in protein structures, which is of course taught by Professor Delineo, we talk about looking at membranes, and you can like get structures of them. And I was curious if you guys found any sort of structural information which really helped delineate between, okay, we know the membrane is different than, say, the regular wild type in this way, which then therefore would then lead to our experiments failing. And I was curious if you guys did any sort of like structural determination. So like you could do a cryo-EM and essentially get a membrane topology and figure out what your structure actually looks like of the membrane itself. And then from there, troubleshoot what's going on. I was curious if you guys did anything like that.
No, we did not do anything like that, but that's really interesting. I think that would be a cool thing to do. We know basically, I think, what that membrane composition might potentially look like. So it'd be interesting to see if that lines up with any sort of like imaging. But yeah, that would be super cool. But no, we have not done that.
Awesome. Well, yeah, great job, guys. I really appreciated how you guys highlighted the depth of research as well as the highs.
That was great. Any other questions? No? Dr. Delino.
Yeah, hi. Yeah, great talk. Yeah, just like Ethan said, there's a lot of lows in research. You don't always get results right away. With regard to the transformations with your work, Vanessa, did you consider other types of transformations like chemical transformation rather than electroporation?
I believe this is something that Dr. Gonzalez has done. I think he has tried to do chemical transformations, but the problem is that, as I recall, the chemical competent cells were not competent enough to take up that DNA. But beyond that, I'm not totally sure what has been attempted in the past. That would have been before my time and already ruled out, unfortunately.
So yeah, one of the reasons why we do electroporations in the first place with linear DNA is it's just so hard to get the linear DNA into the cell itself. And so the electroporation just provides us a little bit better than chemical competence to begin with. And even when we have successful electroporations with linear DNA, we get really, really low transformation yields. And so again, you know, just chemical transformation, we don't have, it's not competent enough to take up that DNA.
Okay, great. Yeah, thanks.